Hello, everyone. Welcome to the webinar on scientific advances in the assessment of organ viability. I'm Camille Nelson-Kotton, and I'm the Clinical Director of Transplant and Immunocompromised Host Infectious Diseases at Mass General. And it's my pleasure to introduce Dr. Peter Reese, who is the organizer of this series of webinars. Peter directs Vanderbilt Center for Transplant Science. He is an MD, PhD. He is a transplant nephrologist, and his research aims to improve access to transplant and the care of solid organ transplant recipient with tools from epidemiology, ethics, and policy. His scientific work includes co-leading pioneer trials that demonstrate the safety and efficacy of transplanting organs from donors with hepatitis C infection into uninfected recipients. For his contributions to transplant science and ethics, he received a Presidential Early Career Award for Scientists and Engineers at the White House and was elected member of the American Society for Clinical Investigation. Dr. Reese is a past chair of the Ethics Committee for the United Network for Organ Sharing, UNOS, which oversees U.S. organ allocation and transplant regulation. His mission accomplishes energetic support of talented and motivated young researchers at all stages of training. We also have with us two excellent speakers. The first is Dr. Ciara Shaver, MD, PhD, who is an assistant professor of medicine and of pathology, microbiology, and immunology at Vanderbilt University Medical Center, and is the associate director of the Vanderbilt Center for Transplant Science. Her basic translational and clinical research focuses on mechanisms of acute lung injury in organ donors, lung transplant recipients, and critically ill patients. More recently, she has focused on rehabilitating damaged donor organs for transplant using human porcine cross circulation. We also have with us Dr. Matthew Bacchetta, MD, MBA, who is a professor of cardiac surgery, thoracic surgery, and biomedical engineering, and the surgical director of the Vanderbilt Respiratory Institute. And he directs a research center that is focused on creating organ support systems that provide extended physiologic support for injured organs, bioengineering platforms for organ recovery and regeneration, and developing artificial cardiopulmonary assist systems. Their group is the first to develop a large preclinical cross-circulation platform for extracorporeal support of human lungs rejected for transplantation. Great. And so we are very excited to have everyone here with us today. I will soon turn this over to Peter. I would like to ask that if people have questions, and we're happy to have questions in the Q&A, please put them there. Please don't put them into the chat. Thanks a lot. And without further ado, Peter, we'd love to hear your introduction.
Thank you so much. I'm really thrilled to be here. I'm very grateful to the groups that made this happen. I'm going to talk about the Lancet, but I think it's amazing to see TTS and ESOT and the AST collaborate behind this webinar. And this webinar really initiates a series of 3 webinars that are tied to 3 articles just published in the Lancet. And the Lancet reached out to me 2 years ago now. They wanted to commission a clinical series on transplantation. I think that what they wanted to hear is a vision for where transplant is headed. And I think what's exciting, and I'm excited to make this assertion right now with all the new trainees in the hospitals across the country and the world, is that the transplant field is really in the middle of a major transformation enabled by technology. So I think we have an opportunity to provide a really positive vision for the scientific enterprise of transplant, which is something that, at least in the US, with some new barriers to the advancement of science, a positive vision, I think, is really what we need. So in thinking about how solid organ transplant is being transformed, in a way, we can almost look back to the very first successful transplant at the Brigham in 1954. They had to find identical twins, and that's what it took to get a solid organ transplant to happen. And unfortunately, very few of us have an identical twin. And of course, many of us don't have a sibling who wants to give us an organ. So right from the beginning, you could see that you had to thread through the eye of a needle many things that most people aren't fortunate enough to have. But right at the beginning, we also saw lack of organs. We also saw this tremendous immunological incompatibility problem. And it sort of buried beneath those two, a problem of fairness. And I think those 3 dilemmas have really stayed with us. We need technology to help us grow the supply of organs and get them where they need to go. And then number 2, once the transplant enters your body, It's not from an identical twin, and there's just a tremendous burden of toxicities that create kind of a new disease state for the recipient throughout their life. And then finally, if we look across the globe, the access to this technology is very unequal. And those are the three themes that this clinical series has tackled, and that the Lancet Commission will continue to work on for several years going ahead. So the purpose of this webinar is to talk about scientific advances in the assessment of organ viability and our ability to bring organs to where people are. So I think if we look at slide 2, I just want to touch base on the goals of the webinar, and then just think about the complexity of the status quo. So the organ shortage really continues to limit transplantation, and buried in this are a need for really advanced assessment techniques to figure out how good an organ is, and then to improve the organ's viability to optimize the use of the organ. So if we look at the next slide, I think because this choreography of transplant is so complicated, we often lose sight of the different pieces of the puzzle that I think are about to come together. So in addition to finding an organ donor where the family is willing, we have this very difficult problem of figuring out, is this organ viable? Not just at that moment, but after it gets transported. The organ has to get to where it's supposed to go. We have to figure out where that's going to be, which in a vast country like the United States can be thousands of miles away. There's procurement, there's storage, transportation, and then the surgery. So you have these different pieces happening at the same time. Where is the organ? How good is the organ? Will the organ be good after transportation? And will the organ function when it suffers this, ischemia reperfusion injury? So I think that when we think about the complexity of that problem, and really the dozens, sometimes or hundreds of people, if you think about techs and people running labs and people flying planes that are necessary for this choreography to come together, it's easier to understand what feels like a scandal, which is the large number of people who don't get a transplant and the large number of organs that don't make it where they're supposed to go, or the number of recipients that are disappointed because the organ transplant didn't happen, or when it happened, it didn't go well. So much depends on not only is there an organ and is it good enough, but can we get it where it needs to go? And so I think part of what we can do is imagine a future world where there are enough organs, we know their quality, and we get them to the right person, and think about the technologies right now that are helping us to cross those bridges. So if we look at the next slide, I just want to make the point that when we think about access to transplantation and how much we're not meeting the need, we're only briefly going to get into this in this presentation, but it will come forward, particularly in seminar 3. Across the globe, the need for transplantation is really vast. And the inability to get transplants to the people who are needed exists both within countries and across countries. So many nations don't report their transplant activity, and there are nations that have no transplant activity. But what you can see with the color coding here is that the pace of transplant, the transplant rate per million people varies widely. And so I think the ultimate goal is that you would have a transplant rate that's more uniform across countries, and that when we implement these new technologies, we're able to kind of see where they land and with what level of success. If we go to the next slide, I'll just briefly remind people of the thousands of people who die on the waiting list every year. Across every organ, we see this. And unfortunately, we just happen to be showing the data in the United States and Europe. There are many organs that are discarded. And we know that that's the case in every nation that has an established deceased donor organ transplant system. If we go to the next slide, what we can see basically is that within that pool of organs that are discarded, I think we can think of it as a loss, but we can also think of it as an opportunity that these new transformative technologies will eventually allow us to realize. So I think with that, I want to pause, and I just want to say that it's really wonderful to have two of the most innovative people who I know, Matt Bacchetta and Ciara, talk a little bit about the new technologies that are coming to address this problem. And I just also want to acknowledge other people, Andreas and Adam Griesemer, who contributed to this first article in the Lancet, and it gave us many of the ideas that we're going to discuss today. And with that, I'll take a step back. Thank you.
Great. Thanks very much, Peter. So now we will start the discussion. And again, we're happy to have questions in the Q&A box. So Ciara, could you start us off? Could you tell us more why is there an organ shortage, and just elaborate further on that?
Yeah, so I think that we're fortunate that there are a lot of patients and families who are interested in organ donation, but many of the donors come from deceased individuals who had acute circumstances of their death leading to organ damage. So sometimes it's as simple as we have willing donors, but the circumstances led to organ injury where the organs are felt to be not suitable for transplant. And what's difficult as a transplant organ assessment person is it's really difficult at times to tell which organs are okay and which organs are not okay. And the default is, unfortunately, to decline to transplant an organ if you aren't confident that that organ is going to restore health to the recipient individual. And where the opportunity really is, everything from increasing numbers of organ donors, increasing ability to assess organs, increased ability to potentially modify organs. And those are some of the things that we'll talk about today. But the fundamental problem is we have lots of organ donors. We just lack the ability to identify how the organs may be acutely injured. We lack an ability to understand which injuries will repair and which injuries won't. And we have some infrastructure challenges of how to get those organs that are potentially suitable to transplant to the highest number of individuals that we can.
Okay. And what about, there are ways of supporting people prior to transplant, things like dialysis for people with kidney failure, and ventricular assist device for people that need cardiac support. And they can replace some of the necessary function. Can't these tools be used just to keep everyone alive and waiting for a new organ?
They can. I think advanced technologies like dialysis and LVADs and ECMO are all tools that we have to help keep our recipients in the best physical condition that we have. But all of those life-sustaining technologies and tools carry their own risks and their own inconveniences, where organ transplantation offers considerable advantage to the potential recipient. So they're fantastic tools to buy us the time to find appropriate organs for as many patients as we can. But even with those tools, huge numbers of people pass away with end-stage organ injury that we're not able to support long enough on those tools and technologies. So they're a great stopgap, but they don't negate the need for additional organ transplantation.
Thank you. Matt, could you tell us more about how you know if a donor organ is suitable, and what that evaluation is like?
Yeah, that's a, that's a question that I think the answer is evolving. What we thought was a good organ 20 years ago is very different than the way that we look at organs today. A lot of that has to do with expanded donor criteria, and was already touched on. The utilization, for example, of hepatitis C organs, something that we would never have considered 20 years ago, and that's helped expand the donor pool. But of course, there's specifics to each organ, but there's 4 general categories in which we look at for organ assessment. The organ function, obviously, and that'll vary, again, how you do that assessment by organ. Tissue injury, the visual appearance of the procuring surgeon or someone else who's assessing, whether it's radiographic data or echo data, so forth, and of course, donor characteristics. Now, specifically in lung, when you're looking at organ function, and I'll speak to that not just because I come out of the lung space, but it's easy for us to get an assessment of gas exchange. For example, we can take a look at a P to F ratio. We can get a sense of the airway resistance by looking at the lung compliance. So those are really important features for us to look at in the lung transplant world. Whereas a liver surgeon is going to be looking at, AST, ALT, lactate. They're going to, very critical in that space is looking at, for example, steatosis. So there are very specific organ function parameters, for example, creatinine and kidney and so on, that are critical. We also want to get a better handle of tissue injury. And I think, again, as I said earlier, all of this is evolving. What we did 10 years ago, 5 years ago is, I think, different than what we're doing today and what we hope to do in the future. In lung, you do bronchoscopy that has we can take a look at tissue injury from there. Also is visual experience or appearance, is there basically pus in the airway? Does it clear easily? Does it well up? The liver surgeon taking a look at and getting a sense of congestion of the liver, just palpating it and so forth. And donor characteristics, again, is something that I think all of us are modifying what we consider an acceptable donor. It's certainly different in the thoracic transplant world, I think, really across the board. I will also note that the way that the organs are assessed will vary significantly from country to country, as well as program to program. We can see that there's an enormous amount of variability, especially if you take a look at renal transplantation and the acceptance rates between different centers, they can vary substantially. But with respect to donor characteristics, age is something that we all look at. But I can also tell you that we've modified it significantly. What we would have considered an acceptable donor from an age standpoint, again, has changed and is still evolving. Other things that matter, of course, are the history of that patient. In lung transplantation, we're going to look at things like smoking history. Other organs are going to look at things such as longstanding hypertension, diabetes, all of which can affect end organ function. And so that's obviously very critical.
Thanks a lot. I hear you that it's complicated. And I think every step of the assessment is really important. And we don't really know until, you're in the operating room procuring the organs. And I think that I always tell patients it's like a handmade, each one is like a handmade suit, right? You're going to have to make sure it's perfect when you finally decide. Ciara, can you tell us more about what has been done in recent times to use more organs? I know there's a lot of work that's been done, but could you kind of summarize that?
Sure. I think looking at figure 2 in the first of the 3 manuscripts is really helpful to illustrate this. So when organ transplantation began, physicians and others tried to pick the best organs available, starting, like Peter mentioned, with identical twin transplantation. But it was very clear that that was not enough organs. And so what I think we've already done, like Matt mentioned, is extend the donor criteria that we think are suitable. So with experience taking care of transplant donors and transplant recipients, we've moved to using donor or organs from older donors, for example. And with the advent of some machine perfusion technology, one example is ex vivo lung perfusion, where you can take an organ outside the body, support it with a machine. That was really an innovative tool that let us assess organs over a longer period of time and assess organs outside of the brain death environment. And that led to some increase in organ utilization, which saved many lives. But still, that was insufficient. So in the last 5 to 10 years, we've really seen an advent of donation after circulatory death, where these are heart-beating organ donors who have acute illnesses that are not sustainable, where a family chooses to change goals of care, and that presents an opportunity for organ donation. And donation after circulatory death has led to a new pool of organ donors that were just not considered at all in the past. And again, as a testament to the innovation of the transplant field, that led to a whole new field of organ donor perfusion and normothermic regional perfusion, which we'll talk about later in the session, where new technologies that extend organ assessment and organ support into new arenas have helped further increase the donor pool. And that's where we are now, but we all know that there's a lot of potential of how do we get to sufficient organ numbers. The kidney folks have really led the way in terms of epidemiologic assessment of which donors and which recipients and how to best select a kidney for a specific individual and using data modeling to know where they'll see the most success. The other organs have tried to use those similar methods without quite as much integration into the clinical care yet. So it will be very exciting to see how that improves. With advances in machine perfusion, we have an increased opportunity to assess organs and figure out which aspects of organ assessment are most predictive of organ success after transplantation. And there's a ton of research and many people around the world really working on this, and it's exciting to see this specific area really taking off. And as the machine perfusion and other methods of organ storage and assessment get better, we really have an opportunity to modify organs prior to transplant. So we're starting to see reports about, elimination of viral infection, pre-treatment of organs with antibiotics, and those sorts of things that can really limit some of the risk to the potential organ recipients. And so these are just a few examples of a whole bunch of innovative thinking in the space. And then I think more recently in the last year and a half or so, we've seen the beginnings of xenotransplant as another opportunity of where to find organs. So if we don't have sufficient human organs to meet the need, is the use of animal organs an opportunity to extend life for some of our patients. And so we've seen some success in the kidney, heart, and liver spaces with xenotransplantation that has been very exciting, and I think is a whole different avenue that is coming in the future that may help us address some of the gaps in need. And so I think there's two ways that you can look at the organ shortage. You can see it as a disappointment or a lack of our ability to help our recipients and then to give a second life to our donors. But when I think about this, it's just, there's such a huge opportunity. We have all these organs that are potentially fixable if we knew more how to support organs outside a donor in the most physiologically beneficial way. And I think that's what our series really details in some of the more nuanced discussions in the text. And it's exciting to see experts in all the different organ fields using both similar and different approaches to get the organs to the condition that we need.
Excellent. Thank you for the summary. Could you tell me more about donation after circulatory death and how exactly that works?
Yeah, so sometimes a potential organ donor has a catastrophic illness, say, for example, a major stroke or a heart attack, where the physicians and clinical teams have a low likelihood of meaningful recovery, and where a patient or their family may choose not to continue life support. So when in the past, a patient whose heart was still beating would not be eligible for organ donation in most places. And so in the critical care world now, there's an opportunity to, with a patient and their family, decide not to continue life support. And so in that circumstance, the priority becomes providing that patient with an honorable death in the most comfortable way possible. And that was recognized as a potential opportunity for the organ donor community to further advance the wishes of patients and their families wanting to try to give the functional organs a new life in different people. How the logistics of how DCD works in different varies from hospital to hospital in terms of where that happens, who's present, where the family is, and those sorts of things. But I think the focus is always to provide a comfortable death to the patient at hand, and then move towards donation process to try to extend life for others.
Thank you. Excellent summary. Matt, could you tell us a little more about some of the machines that can be used to improve the condition of organs for transplant?
Sure. The machine perfusion is not entirely new, but the innovation that's been taking place over the last few years is quite remarkable. Sort of the basics of ex vivo machine perfusion are one, temperature. It could be a hypothermic machine perfusion. It could be subnormal thermic. It could be normal thermic. You can use different perfusates. For example, you could use a blood-based cellular perfusate with additives, or you could use an acellular. And some of the platforms do enable functional testing and potentially some therapeutic intervention. I'll use an example of lung transplant. For example, a lung on ex vivo lung perfusion, you can perform bronchoscopy where you may be able to clean out the airways, to assess the airways and so forth. It also allows you to globally assess the compliance, which is really critical because you can have regional differences. For example, you could have an infiltrate in the right lower lobe, and you may see that compliance improves over time or it doesn't. And that is really critical feedback in the assessment of organs. There's been remarkable headway made in the liver space by the Swiss group on the extension or in the duration of support for livers. I think one thing that's really critical for us to recognize is that as good as the machine perfusion is currently, there are still very severe limitations, and it has to do with not being able to fully recapitulate the physiologic environment in which organs sit, meaning in the body. And that is a serious constraint to the ability to recover an organ. And when you start to think about organ recovery and regeneration, you really do need to have that homeostatic physiologic environment. Our bodies will heal. If you twist your ankle will improve over time, but there's this whole process that we're all aware of, and having a homeostatic physiologic environment is what enables that. That's very limited and constrained within machine perfusion systems. However, there is still an opportunity to potentially reduce the ischemic reperfusion injury by utilizing machine perfusion. And I think this is a very active area of research. And, the tale is not done with this one. There's still a lot to be done. I think a huge opportunity for us to recover organs that we never would have recovered in the past.
Camille, do you mind if I make a quick comment?
Please do.
I was just going to say that I think one of the things that's really interesting is the fusion of multiple steps in this complex process inside one platform. So, Ciara was talking about, the different ways that we think about, how good is this organ? And there are a lot of different ways to think about it. You have various people who say, well, I need to put a needle in it. I need to look at it under a microscope. That's a good organ. Other people want to build an epidemiological model. And now, we have these platforms that perfuse and, measure things like resistance. And then, but this is also the same platform that we really thought is, no, the job of this platform is to, enable the organ to be ready, and to be transported, 500 miles. But now, in addition, it's a platform where you do things to the homeostatic environment. So I just think it's really fascinating that everyone had a different idea of what putting a kidney in a box, what the purpose was. And these purposes have really come together in, I think, an exciting and synergistic way.
Thanks. Agreed. Matt, could you talk a little more about storage temperature? And, the classic thought is that things belong on ice, and then where we are now with that.
Yeah. It's a remarkable story that actually started a couple decades ago, and we just never really utilized the research. So we're all sitting around talking about how novel it is, but the research that underpins a lot of the new ideas and paradigms about temperature storage really are date, a couple decades ago. I think what happened, and as we all know, for decades, we literally just, we took the organ, would have it packed in a bag with a perfusate and put it into a cooler packed with ice. And that was what we did for decades. Studies that go back, as I said, at least 2 decades demonstrated that organs performed better when they were kept at a slightly warmer temperature, 10 degrees Celsius. And that was demonstrated in multiple animal models. We just never really adopted it until more recently. And now, of course, you'll find certainly in the lung space and the growing heart space as well. And, kidneys have, many times have, for decades been stored with certain perfusion machines at temperatures that are not on ice, or they're not necessarily included on ice. And that the studies have demonstrated better protection, especially with respect to the mitochondria, and that this has reduced the ischemic reperfusion injury. Organs do not like to be on ice, and they don't like to be cooled down on ice, and they don't like to be stored at ice on ice. And so I think this has really opened up so many opportunities for us, because it's changed the logistics of transplantation in many organs. Many times, I mean, when I started my career, operating in the middle of the night was just what we did. And now we can basically take organs, for example, in lung, and we can extend that storage time out to 20 hours or more. And it gives us the ability to arrange for that transplant in a way that we didn't have before. It also means that we're able to use organs that we probably never would have used in the past, and the quality of that organ is better. The heart space has done this, and there's a lot of studies that are out there right now demonstrating superior clinical outcomes with a temperature that is somewhere probably between about 8 and 10 degrees Celsius.
Excellent. So, Ciara, what new tools are there to help assess organs better, and how do you think that's expanding the organ pool? We talked a little about this, but maybe you can elaborate a little more.
Yeah, I think there's a lot of opportunity, and it varies by organ. I think we have new imaging technologies, for example, that let us better assess tissue perfusion and organ perfusion during storage. There's advances in PET scans and ultrasounds that let you get a better sense of whether tissues may be permanently damaged or have an opportunity for reversal. These are things that right now are used mostly in the research space and aren't yet translated into the clinical world, but are kind of ready and waiting in the wings to be able to do that. I think there's been, in the last decade or so, an explosion in omics technology. So every time you open a journal, you find transcriptomics or proteomics or metabolomics or other biomarker assessment. And there's new statistical tools and techniques to take those data and integrate them, so you can learn from all the different levels of sort of tissue homeostasis and tissue pathophysiology. In the donor space in particular, it's sometimes quite tricky to get the tissues to do those types of analyses, but where they exist, that data has been very informative to give us not just an assessment of what pathways may be disrupted in organs that are ultimately deemed suitable or not suitable for transplantation, but to really understand what's happening, which gives us new ideas for potential new therapies that may work for donor management, but may also be useful in management of the chronic conditions leading to transplant in the first place. I think another area we haven't mentioned at all is how does AI fit into donor organ assessment? And I think that's an unanswered question right now. But as we move forward, we will have more and more information about potential donors and potential recipients and how those might match. And really leveraging machine learning and other assisted decision making can help different transplant centers learn from other centers. I think at the very beginning of this session, we talked about that there's variability both within countries and across the world in terms of which organs are thought to be suitable for transplant. And part of spreading the practice to use extended criteria donors, for example, is really learning from bigger centers that perhaps take a little bit of a different risk in terms of organ selection and learn which organs work well, even though you weren't certain that they would, and then apply that technology. So if you think about it like, a potential exchange program, like this heart would be used in Europe, but not used in the United States. Well, why is that? And can we disseminate those successful outcomes from certain donors in a way that is usable in new areas? So, and then biomarker development is really the last area, that the longer a time you have to assess an organ and assess what's happening at a molecular or pathophysiologic level, the better the opportunity there is to learn which specific biomarker features are most predictive of behavior after transplant. I think with all these new things, what we really need to do, there was a question in the chat about how do we bring technologies like this to really worldwide use. And part of that is affordability, and part of that is dissemination of decision making. So if I can use a fancy biomarker in the United States to tell me that an organ with these clinical characteristics is functional, maybe someone in another part of the world doesn't need the fancy biomarker. Maybe the biomarker can just give us the reassurance that an organ is okay, and we can base further decisions on clinical characteristics that are more widely available. And all this has to be tested and worked through for clinical translation, but the opportunity is immense.
Thanks a lot, and thanks for your emphasis on international equity as well with all of this amazing technology. So when you have these organs, can't you just modify them during storage to make them amenable to transplant? I mean, do you have the power to do that? And Ciara, could you comment on that? And then maybe Matt could follow up with further information along that line.
So we can and we will, but I would say modification of organs in some ways is not yet widely used. So we give organs antibiotics and things like that to try to begin treating the circumstances of a donor organ prior to moving it to storage. We try to use different temperatures to maintain organ viability. But there really is potential to, use UV therapy for viral infections in an ex vivo situation. There's some data about changing ABO tissue typing during organ storage that maybe could take a donor and make it truly a universal donor where you could put it into a specific recipient. You can change animal antigens, for example, to make them more hidden to the human immune system. So you can really, if you can think of it, you can imagine it. And with the advent of CRISPR and other technologies, it really is feasible to genetically engineer lungs, hearts, kidneys, and livers to be less inflammatory and more reparative pathway activations to really try to optimize the health of a donor before transplant. So we're starting to see this, and I think in the next decade, this true modification of organs is going to become more mainstream. I'll leave it to Matt to talk about how to build organs and move those areas along.
Such an easy question. If only I had a magic wand. So obviously what Dr. Shaver mentioned with respect to organ modification, there's a very active research area, and groups around the world, research groups, have made very good inroads into that. For example, the blood group modification, the Toronto group's done some very interesting work in lung to make modify A lungs into O's and so forth, and looking at reducing viral load, for example, in hepatitis C positive organs. They've also looked at, many groups have looked at trying to upregulate IL-10 in organs, and looking at different techniques to do that, to limit the expression to the transplanted organ alone. With respect to building organs, that is, a lot of groups are looking at it, and you may have read recently that the United Therapeutics Group reported on using a porcine liver substrate that was recellularized with human cells as a bridging technology for a patient. And so there's this area of interest looking at patients that may have acute fulminant hepatic failure who, if in the worst circumstances, would need a liver transplant, but there may be an opportunity to bridge that patient to recovery or to bridge them to transplant. And so there are groups looking at creating this decellularized substrate, which means you maintain basically the ultrastructure of the organ, the extracellular matrix, and then you humanize that porcine substrate with human cells. Now, there are some organs that are probably going to be easier to do that in than others. Nothing against my liver colleagues I've got lots of friends who are in the liver space, and I love them, but you know, they don't have as many cell types to concern themselves with. In the lung world, we're looking at, you know, upwards of 50 different cell types that have very specific regional needs and function. And so it's very, you know, the cell type that you see in the trachea is very different than what you see in the very distal airway and in the alveolar space. And those functions of those individual cells is critical to having a functioning organ. So there may be opportunities to get there faster in certain types of organs. I know they're trying to do the same thing with kidneys, and you know, the structure of a kidney is quite relevant to its function. So it's one thing to say I've recellularized the kidney, but if you don't recapitulate that very specific proximal and distal tubules and so forth, and the cortex and the medulla, then you're going to have a kidney that probably is not going to function properly when you're using the decell recell platform. Now, xenotransplantation is a whole different animal, no pun intended. And to share the quip of Dr. Shumway, really a father of heart transplantation, you know, xenotransplantation, he used to say, was the future of transplant and always will be. We have seen a tremendous amount of progress in xenotransplantation. We've seen, now that we have an approved study in the renal space, we have had experience with decedent research. One thing, though, that we have to understand is it requires more intensive immunosuppression, and that even where some of the decedent studies that were done at NYU are out to 60 days, where they seem to have reasonable function, there's still the detection of an ongoing rejection pathways. So we're not quite there, but we've made obviously tremendous, and I say we in the global sense because I don't make xeno organs, but we have made tremendous progress in there. I think that there will be some organs that are going to be an easier xeno pathway, and you can see that playing out currently in the field because kidneys seem to be more of the low-lying fruit. I think that it will be, and hearts maybe as well. I think it's going to be extremely difficult in lungs just because of some anatomic issues as well as some functional issues to make that work.
Thanks a lot for the excellent summary. Such exciting times in transplant medicine, right? There's so much happening and so much amazing science and collaboration across so many different disciplines, and then also between academics and clinical and industry as well. So really exciting times. Can I ask each of you to just answer some of the questions in the Q&A? And for those of you listening, we're happy to have more questions in the Q&A, but Peter, do you want to take a stab at a first question?
Sure. I was just going to make the point that I think one of the things that makes me optimistic is that, if you think about, what do Ciara and Matt and I do? We work for patients who have end-stage organ disease. We serve them. And in the past, what had happened was there was really only, dialysis or a kidney transplant, or, there was, a pump or a heart transplant. And now I think what's really great is that you have multiple competing technologies and they're each getting better. And so I think one of the things that will happen for us in the future is that we may go beyond being transplant doctors to being doctors who are standing sort of at a switchboard where, it's funny to say this, but, if you could make an incredibly successful heart pump, maybe you wouldn't even need to do a heart transplant. If you could make an incredibly successful xenopig organ, maybe you could walk away from deceased donor kidney transplant. And ultimately, no one would be happier about that than me. Because all that we should really care about is the extension of life and the quality of life for these patients with end-stage organ disease. And what we don't want to be stuck with only one potential lane of innovation, but instead you can have organoids, you can have decellularized, recellularized platforms, you can have xeno organs. What we have now, I think, is like a tremendous amount of innovation that we're really focused on in the deceased donor organ space. And I think it's been great. And I think in each one of these spaces, incremental growth, you just add it together and then hopefully you see like real relief of the organ crisis. Now, Camille, I just wanted to tackle a question that you and I could answer together, which is, someone was asking about donor-derived infections like HIV. And I think that that's another, I think, really interesting area. We saw tremendous success with organs from donors with hepatitis C cure. With HIV, you sort of see a different set of innovation where you had kind of legal and regulatory innovation. It was illegal. And then we had, ethical and imaginatory innovation. We were so scared of the stigma of HIV that it was unthinkable. And then, we had sort of innovation along the lines of just controlling infection. And I think you're a lot closer to those trials than I are, but that's one way that there was kind of incremental improvement, but we had to open our imaginations, build a regulatory framework before we could even do the medicine.
Right. And I do think it's time to think about that, pushing the envelope, thinking about hepatitis B, surface antigen positive donors, potentially HIV positive into HIV negative. We have amazing treatments for HIV these days. So it is time to begin thinking about those things. I share your comments, about and concerns about things like social stigma, which is unfortunately still present. But we deal with patients often with life-threatening illness who really need transplants. So we really need to think about how we are going to expand the organ pool as quickly as possible. The HOPE Act allowed us to understand that roughly half of all donors in the US who are believed to have some type of markers that they are HIV positive are actually not HIV positive in the long run. So that's a pretty amazing experience. And I do wish we could open that up, especially to critically ill patients who might actually die without these offers. But we're not quite there yet, unfortunately. Ciara, Matt, have you picked a question from the Q&A you want to tackle?
And there was a question about which of all the things that we're talking about has been most important in driving increased transplantation. And I think that's a tough question to answer, but I think what I've learned personally in watching and participating in transplant programs growing is we need to do a better job of learning from others. So the way that extended criteria organs sort of came into clinical practice was adventurous recipients and adventurous transplant physicians said, I think this organ might be okay if we transplant. I know we don't usually do this, but I have a patient with end-stage organ disease in front of me, and I think this will work. And it's a team effort between the clinical teams and the patients and their families to take what might be a risk as part of a clinical trial or as part of a new innovation in the transplant space. And when we have tried that with things like hepatitis C, where we used to just think, well, that would be just awful to do. As technologies improve, some of that risk gets lower, and you try it, and then when you see those successes, you can take the anecdotal successes from one or two patients and build those into trials and dissemination elsewhere. So I think there's a hefty dose of courage in part of bringing innovations from the scientific world into the clinical space, and trying to do that with the patient really at the center of that to try to save more lives is really where we can bring that. But I've certainly learned a lot from colleagues from other centers and colleagues around the world in terms of learning from practice variability, really what is possible and what can be done. I think that's an easy, not very expensive step that can really happen anywhere in the world.
There is a question in the Q&A session asking about ex vivo perfusion for lungs and whether or not we use normothermic or hypothermic machine perfusion. We use normothermic. The major differences are whether you're using a cellular or acellular perfusate. We use an acellular perfusate, which is based off of a Steen solution and the Toronto protocol. The question about how do you assess lung perfusion? Currently, we take a look at things such as the pulmonary vascular resistance, and that gives us some feedback on how well that organ is perfusing. The gas exchange efficacy is done basically through oxygen challenges. You can take, the ventilator from 40% to 100% and get a sense of how well, what's the delta on that gas exchange, meaning that how well does the PaO2 improve. In the there are other groups that use things such as ultrasound on the lung to take a look for areas of edema. In our laboratory, we do things like thermography to look at the differences in how well different parts of the organ is perfused. I don't want to spend too much time on that unless you want me to, because I can talk about that all day long because it's an area of research for us. But hopefully that answers your question.
Peter, do you want to tackle another question?
Sure. I'm thinking about, so one of the attendees asked about the U.S. government's role in organ donation and transplant systems, whether it's created problems and limited innovation. And some of the, I guess what I would call unintended effects with having standards. And, it's interesting. I think what I would say is that we really need a transplant regulatory environment that works well with transplant centers. And I do think that the transplant regulators have the best interests of the patients in mind. I think making transplant policy successfully is very hard to do. One of the things that emerged from this clinical series for me is the realization that a very good data collection system is totally fundamental. A national rigorous one is totally fundamental to the success of transplant. It's necessary because, and there are a lot of countries where they don't have a national system of data collection. And so one of the things that's at the foundation of our field, we really take it for granted in the US, is that we're a trust-based field. We're a human generosity and reciprocity-based field. And once you have any major widespread concerns about trust, reciprocity, transparency, professionalism, organ donation rates can go down, and this incredible coordination infrastructure that I was talking about at the beginning can fall apart pretty fast. You saw that actually fairly recently, a couple examples in Germany. There were some hospitals that were falsifying the urgency of the patients on the waiting list, and deceased donor rates went down. And I think that there was also a kind of a bad story that emerged out of a hospital in India recently as well. And so I think we, even though regulations can be burdensome and they can inhibit innovation, we really need this investment. We need this policy because it maintains trust. So I'm hoping that things get better in the United States with the function of the OPTN and the faith in the OPTN. And I guess my main thought is that maybe we should hope for a system where policy revisions happen faster. I feel like we've been stuck a little bit with transplant with like, it takes a long time to make policy, and then once it lands, it kind of sticks. And maybe what we need is like faster rounds of refinement, because it's a complex system. And when you have a complex system, unintended consequences are probably to be expected.
Ciara, do you want one more question? I think one more question for you and Matt to each answer, and then we'll wrap up.
Yeah, I think one of the questions was related to, we talked a lot today about deceased donors and DCD donors, but there was a question raised about living donors as a potential tool. And I think really we look to our kidney colleagues for having really led the development of that whole new field, honestly. So kidney and liver, less so lung, and obviously not a heart, but there's huge potential in leveraging living donation to provide working organs, working lobes of organs, to provide transplantation. I think the logistics of it is tricky, and also making sure that we're protecting the living donors. Peter can speak more to that, but there's a huge opportunity, and we don't want to imply that is not at all a valuable addition. It's a massive benefit to the transplant field, and doing transplant chains where one person may donate into the system to then cascade through and benefit other people is something that has saved a lot of lives over the years, and is really exciting to see. And that gets back to Peter's comment that organ transplantation exists as an opportunity because of the generosity of human beings in the worst situations of their lives, and looking outside of their own circumstance to see how they can better the world and better the lives of other people. And I think that's an amazingly hopeful and encouraging opportunity. And we're the lucky ones that get to work in a field where there is such innovation and such opportunity to not just learn new things in advanced science, but to really get to do that while saving lives at the same time. And that's really fantastic.
I know we're short on time. There's a question here about how far are we from intercontinental exchange of organs and their repair in organ repair centers. I'm going to say that this is mostly a regulatory question because, for example, we can literally keep lungs out for 24 hours. We can utilize machine perfusion systems on kidneys for more than a day, 2 days, and so forth. And basically, you can get to most places in the world in 24 hours with implant for those organs. But whether that I'll kick that one to my colleague, Peter Reese, because he does a little more work on that regulatory side. But I mean, we could literally do this tomorrow. We could be sharing organs on an intercontinental basis tomorrow.
I mean, I guess all I would say is that I think, regional organ repair centers, it seems like there are some suggestions that that's the direction that things are heading. My colleague, Emily Vale, and others have been working a lot on, sort of understanding donor care units, where they should be physically located, what attributes they should have. But I think these donor care centers, they also are another place where you could imagine different things happening at once. Therapeutics, trials on donors, the availability of more intensive technologies to evaluate organs when they're still in the donor. I would love to see that kind of cross-border collaboration. We have seen some of our high-risk organs flying up to Canada, mostly because there sometimes U.S. centers aren't willing to take them. So at least there are some innovators out there who've been thinking the same question: why respect borders when collaboration can cross the border?
Thanks very much, everyone. Great discussion, amazing publications. So this is the first of 3 publications. They're all out in the Lancet as of this past week. And we will have 2 more webinars discussing these terrific papers. I hope everyone finds them really useful. For me, they're great reference works as well, and sort of really where are we with a very modern transplantation. So thanks a lot. This will be recorded and available through TTS and some other websites as well. And thanks a lot to the participants and for your amazing work in the field.